not identified and not modeled until James Watson and Crick came along with the help of Rosalind. Um, ah, what's her name? Franklin, right? Rosalind Franklin in the 50s. So I added this slide. This is showing that DNA is also in bacteria. A single bacteria cell can have DNA. There's no nucleus there, but you can see all this. It's almost always in almost all bacteria one circular, a little twisted up, but one circular strand of DNA inside bacteria. And that bacteria has many, like we talked about 2 weeks ago, has many structures. Many of the parts on it are the exact same or very similar to what you see in humans or other living things. All right. And then there's these tiny items called plasmids, and those are just floating little DNA things that might have some level of um, uh, capacity for that bacteria to develop um, a no to resist a resistance to a certain drug or something like that. So if there's a resistance to a drug that may be that a bacteria develops, that may be kept right here in these tiny little plasmids. Chromosomes in a chromosome in a bacteria also doesn't have a pair like there are in most animals. There's pair chromosomes, and it goes through the process of meiosis where it splits up, and you get some chromosomes, some genes from your mother and some from your father. But this is a single chromosome in a long circular rounded thing that just passes on, and so they go through slightly different levels of reproduction. So we talked about the 23 human chromosome pairs, and there you can see number 2, which is actually 2 different chromosomes in all other great apes. And other than that, there's very similar structures on all the rest of them. You can see similar items, similar information kept on similar chromosomes in similar areas. Okay. So I wanted to go through this because I knew some new people would be here and maybe some new people would be listening. Added a few different ideas, but this is somewhat of a review of the first part of my talk. Um, then we talked about the DNA and how it has always this sugar phosphate backbone to the DNA. And then there's the different base pairs. Adenine always combines with thymine, and guanine always combines with cytosine. And so as these build up, as these structures build up, these nucleotides build up, it makes a long every we said every chromosome inside animal cells can be up to 6.5 feet long of information kept in every cell of the 37 trillion in your body. So fascinating stuff. All right. I'm going through this kind of fast because this is stuff we talked about. This is one I added on, a little slide I added on just to kind of look at it a different way. This kind of looks closer and closer and closer at the steps in reproduction. But here's here's a chromosome that could be an immense length, and this is highly magnified, of course, but a certain section of that, which could be maybe 400 base pairs long or up to a couple thousand base pairs long, would be a gene that is coded to do something inside your body. So there would be a gene here, followed by another gene. There could be a whole bunch of areas that used to be called junk DNA, but now it's more favorably thought of as unknown what its functions are, or we're trying to still study and understand what their functions are. So this DNA molecule then, if you look at just a little part of one little gene in here, it can have a pattern similar to this. With the strand of DNA can have these different nucleotides, each paired together, or each here. And this is assuming that it's after it's split apart in the process of reproduction. All right. Then inside the molecule, inside the nucleus, it goes through a process of what's called transcription. And transcription means that additional nucleotides, the ones that match up, are formed and are bonded on back onto these in a way that it produces an RNA, messenger RNA molecule, which has the opposites, the complements, I should say, of the different nucleotides. So if this one has a CAG, a proper transcription of that for the messenger RNA, it gets the energy to make a G and then a U and a C. U is uracil, which replaces the thymine on the messenger RNA. So this is a reproduction of the complement of this, right? And I was looking at a number of interesting videos. You can look online with the Khan Academy or different things like that, and you can see some really nice videos that show how this transcribes and the method by which it transcribes. There's information that tells the messenger RNA to start building at this point, and fascinating kind of microbiology of what happens as these uh, DNAs reproduce. So, oops, hit the wrong button there. So that messenger RNA then works in triplets, as we talked about, and the triplets code for one of the 20 different amino acids, okay, that are in the building blocks of life. So this makes GUC makes a certain amino acid. Then the UAA codes for another amino acid. And these amino acids, these long strings of amino acids get built together. And those are called proteins. When you have a long string of amino acids, it's a protein that codes for this protein to cause certain things to happen in your body. It's phenomenal when you think of it that it happens. Okay. So that's the basic kind of premise of how it